All right, thank you everybody for coming and finding your way here. A little bit different pathway. Apparently, I'm guessing that they've shifted to the opening now to the east, so you should be able to come in and get to the front door, but come in from Rights Union side. Don't start your way down there, otherwise you'll have a circuitous route like several of us did. But I'm happy to introduce our speaker today, Dr. Christopher Dutton. Chris has been a postdoctoral associate at the University of Florida Anthropology and Biology Department since 2019. Dr. Dutton is an interdisciplinary scientist working at the nexus of ecology, bioinformatics, biogeochemistry, microbiology, and low-cost open-source technologies, where his research focuses on how human and non-human animals and their microbial symbionts, microbiomes, interact with one another and with the environment. He received his master's in environmental sciences from the Yale School of Forestry in 2012, and his PhD from the Department of Ecology and Evolutionary Biology at Yale University in 2019. And with that, we are going to hear about a megafauna that's not common to North America unless you go to the zoo, so it should be really exciting.
Yeah, great. Thank you very much. Yeah, I'm going to talk to you about some of my research on hippos and kind of how hippos can alter ecosystem functioning in the environment. And I call this talk kind of the vanishing because, you know, I would argue that hippos were in the past across most of the landscapes of Europe and Africa. But even here in North America, there were hippo corollary animals that had a very similar life history. So when I talk about these effects that I'm going to show you that are occurring in a river very specific in Kenya, just imagine like the river just down the street here that maybe it used to look like this.
That wants to get control.
So animals can shape the environment in lots of different ways. You know, we all know about trophic cascades and predator and prey interactions and the general movement of resources around environments. And these can have alter how these environments are functioning. So
you might have to click on the screen.
got it. I'll just use a keyboard. So it's pretty well established animals influenced by geochemical cycling. You know, this great paper by Schmitz at Yale a couple years back showing the myriad of ways that animals can influence the carbon cycle. And so I'm particularly interested in the role of animal subsidies. So a subsidy is when an animal brings a resource from one ecosystem and deposits it into the other ecosystem. And the one thing I really want you to take away from this is that it is context dependent. The way that subsidy affects the recipient ecosystem. I'm going to talk about the context dependency a lot. And these things have been well studied in the salmon system and waterfowl and alewives in North America.
I'm sorry, I'm just gonna check one more.
That's okay.
YouTube is streaming, just not your stuff. So you're sharing. It is streaming, screen sharing. So I think we're just gonna have to go with it. We'll post the recording from Zoom to YouTube later.
Okay.
What's cool about all this animal ecosystem work is because then we get these really great modelers like Dowdy and others who then try to reconstruct the past. You know, how did ecosystems used to look prior to the extirpation of megafauna? And generally, they kind of found that there's been a lot slower nutrient recycling rates, less movement of nutrients between ecosystems, reduced methane emissions, and lots of other ways that ecosystems have changed from how they were traditionally evolving. So our problem is that our current understanding of how ecosystems function are really based on how ecosystems function now, and ecosystems here are pretty much devoid of megafauna, you know, because they've really been extirpated from a lot of the landscapes. So we're missing, you know, potentially valuable linkages between animal ecosystems and their microorganisms, their microbiomes. And why is this important? I think you probably all know this. You know, understanding the role of animals in affecting ecosystems is really critical for quantifying the loss of ecosystem services from continued extinctions, whenever we're trying to reconstruct the past, and also for understanding future trajectories under climate change. Well, Africa is a really great place to study how animals influence ecosystem functions, because in Africa, there's just animals everywhere. And the hippo, their current range is denoted on this map in green, and their historical range in Africa is denoted in red. And so they're in a severely contracted range right now. But again, hippo corollaries were across most of the world, you know, an animal that had a similar life history. but hippos used to be in Europe and Asia as well. So we don't have many left, which is like one of our urgencies of studying the roles that they can affect ecosystems. Hippos are one of the largest land mammals on the earth. I think only the elephant and the white rhinoceros is larger. They're very, very dangerous of an animal. If you don't know about kind of what they do, during the day, they kind of hang out in the water. You know, they like to prefer these like slow moving pools with a nice soft bottom for their feet and protection from desiccation from the sun. Hippo pools are really great systems for studying the context dependency of these subsidies, because you can see hippos in this like really long stretch of river, really spread out. But then we also get hippo pools like this that are, you know, like over 100 hippos really in a tight congregation. And so the effect of the subsidy is going to be very different between those two environments. In the evenings, hippos leave the pools. They go and eat on the savanna. They're like a moving lawnmower, just constantly eating all throughout the night. What's interesting is occasionally they'll go and they'll mark a tree or a bush or a rock with their feces, kind of like a rudimentary GPS point, you know, if you can imagine it during the evening. So it's always easy to find fresh hippo feces in the morning. But with that said, most of their egestion occurs actually within the river. If you've never seen hippo feces before, it kind of looks like this, really coarse particulate matter. It's just kind of like when you've mown your lawn at home. It's, you know, it looks like just like grass clippings, basically. So one of the first things we did was try to quantify the amount of loading that the hippos are bringing into the system. So in the Mara River of East Africa, we work in about a 100-kilometer stretch of river, and there's about 170 hippo pools in that stretch, and there's about 4,000 hippos. And the context dependency of the loading really depends upon how many hippos are in each pool. And so that's just kind of what this figure shows. The different colors indicate low, medium, or high, or very high levels of total nitrogen, total phosphorus, and total carbon that are going into those pools. So you can see it's very kind of distributed throughout that. And it, so there's about 8,500 kilograms of organic matter per day. For reference, that is about the weight of an African elephant. So there's that much organic matter going into the river. If we break that out by carbon, there's about 3,200 kilograms of carbon per day going into that river. And that equates to about a dump truck and a half of carbon being dumped into this river every day. Over 1 week, that is over 11 dump trucks of carbon. There's just a massive amount of resources going into this river from this hippo population. So we first tried to understand what are the overall downstream effects of hippo loading at that whole river level. We installed a water quality sonde downstream of all 4,000 hippos, and we found something super surprising, that hypoxic floods were fairly frequent in the system. So on the bottom is discharge in black. So every time the discharge peaked, that there is a little flood event coming through, the dissolved oxygen would crash down. We recorded about 49 of those events. 13 of them were hypoxic, very stressful to the aquatic life, the fishes, the macroinvertebrates. And we recorded 9 fish kills over 5 years from these events. Not all floods are created equal. This just shows you the magnitude of the drop and how long it took after the minutes of the beginning of the flood, to give you an idea. We don't have a really good understanding about how this functions throughout the entire river channel, but it corresponds to where it is raining and how many hippo pools are flushed and how much chemical and microbial action we have going on that crashes at DO as that wave of water moves downstream. But the fish kills have been extremely difficult to document because this is the Mara and Serengeti ecosystem. As soon as something dies, something else eats it. And so we think that there's been many more fish kills than what's been documented, because if you didn't actually see it happen, if you showed up the next day, all the fish would be gone, because it's a great resource for the scavengers in the system. We were able to catch a couple graph samples from these events. The discharge is at the top. So as discharge goes up, dissolved oxygen crashes, specific conductivity goes up, ammonium goes up, biochemical oxygen demand goes way up, and then hippo feces flux goes up. So when I was at Yale, I developed a sediment fingerprinting procedure using some Bayesian algorithms, and then where we can actually determine the amount of hippo feces within a piece of sediment within the lower river. And so at the peak of this flood, there was 100,000 kilograms per hour of hippo feces coming through that river system. That's a massive amount of material moving through that river. And if we do the math on that, that only equates to about 12 days worth of loading for the hippos within that river. So it makes sense that we could get that much. I'm a big experiment person. We like to try to experimentally induce some of these effects to try to understand the mechanisms behind it. So we have a little experimental stream array that we use in Kenya with these flexible PVC sheets and some galvanized steel on the sides to keep it upright. We've got a little paddle wheel that kind of turns and moves the water around the circle. So it's just a recirculating stream that we have. We've got 12 of those in Kenya. So what we did is I went to a hippo pool, one of these really high BOD hippo pools, and I pumped water out from the bottom of it with a peristaltic pump. And as you can see, the top of the hippo pool is brown, but the water being pumped from the bottom is black. You know, it's extremely like high in reduced compounds and anoxic. I then spiked that into these different streams in different amounts to try to understand what would that do to the dissolved oxygen. We also had a dissolved oxygen meter in each of the streams. And so this is the control treatment, and we do the spike at the blue arrow right there. And so what we found is that the more and more that we spiked into it, created an increasingly larger response in the crash in dissolved oxygen. And so, you know, this was super interesting. It's likely more of a chemical process, something that goes on that fast, rapid oxidation of those reduced compounds. But it was also anoxic. And so we calculated, well, just by the mixing of anoxic with oxic water, how much would we expect it to drop? And it would have fallen along that line. But we actually induced a much larger drop in dissolved oxygen, rather than just the mixing of the anoxic. And so, you know, this is more of a chemical process occurring, we think. We also took hippo feces of different amounts and spiked that into another set of our streams and did the exact same thing. So here's the control treatment. And interestingly, what we found was a different effect of hippo feces. So it goes down, but it does not recover, and it keeps going down over time, which is indicates like the respiration of carbon. So there's some microbial action occurring within that stream. It's hard to extrapolate small little experimental streams out to an entire bigger river system, because you've got different surface area and different types of dynamics occurring. And so we did a stagnant film model to try to understand what would have happened in the main river if we had put that same proportion of hippo pool water into it. And so this is the reference, the control is in green, and the 10-liter hippo pool water addition is what happened in blue. And when we extrapolate out to the main river system, we actually get a much larger increase in the drop of dissolved oxygen. So again, giving us good evidence that you know, this hippo pool water flushing through the system is really driving some of these events. But we really needed more evidence, you know, scaling up experiments. So what we wanted to do is flush an actual hippo pool, but hippos are very, very dangerous. So what we did is we went to a pool that had never had a hippo in it before, and because we know some Maasai, we have some friends over there, and so they could tell us there's never been hippos in this pool. We built a dam upstream of it with a whole bunch of sandbags. We let the water build up over several days. We then rapidly breached the dam so that we get a flush through there, like a small flood. And this doesn't look very impressive, but it's like really impressive out in the bush when you're carrying all the sandbags and stuff. And then we measured everything downstream that was going on of the pool. We then found some nice enterprising young men in the local town who had this lorry and these large water tanks. And we convinced them to go pump 16,000 liters of water out of a hippo pool into their tanks. And then they brought it over to us, and then we pumped it into our pool. So we're making this a hippo pool now, essentially. We took all that material out of the bottom of a real hippo pool and brought it in here. And we did that over 2 days. We then put that dam back in place and then breached it and measured everything downstream. So this is what dissolved oxygen looked like with that control flush. So it goes down a little bit. So there's probably some organic matter kind of bringing up, or a little anoxic bottom layer at the bottom of that pool. When we did it with our fabricated hippo pool, we got a much larger response. So again, giving us very good evidence that these flushings of the hippo pools are causing these big drops in DO. And you'll see that dissolved oxygen starts a bunch higher because that hippo pool water is just like full of nutrients as well. And so it's, you know, algae is probably responding to that. If we look at biochemical oxygen demand, We also get a much higher response after the flush. So that gave us really good evidence that these flushing of these hippo pools does cause these hypoxic floods. And the mobilization of that hippo pool water and the hippo feces, kind of the combined effect of that is really driving these large crashes in DO that we see. There's a higher degree of dissolved oxygen depletion during the floods with longer intervals between floods, because that allows more stuff to be mobilized and pushed downstream, which has a much larger effect on how the ecosystem functions. And if there's any fish people in the room, any fish person would tell you that it's actually really difficult to tell how a fish died if you have so many coast stressors. But we think it's partially due to the high concentrations of suspended sediments and the other reduced compounds in the water probably contributing to the fish kills. And interestingly, we think this is just a normal a normal functioning river, how rivers may have functioned prior to the extirpation of megafauna. There's no indications that this is different from a normal African river that has hippos in it. We really wanted to go to the specific hippo pools and try to investigate these processes on a very fine scale. So what we did is we found about 17 hippo pools and 2 pools that had never had hippos in it. We then took biogeochemistry measurements upstream, downstream, at the surface of the pool, and at the very bottom under the hippos of the pool. We then measured about 37 different biogeochemical attributes, like all forms of carbon, nitrogen, and phosphorus, dissolved gases, reduced compounds, all major ions, dissolved oxygen, oxidation, redox potential, everything that we could think of, we measured it to try to really understand what's going on. We also took information about the context of the pool, how many hippos were in it, what was the discharge, what was the volume of the pool, how much water could it hold. What we found is that water residence time is really the key determinant of hippo pool grouping. So I could break these hippo pools up into 3 types: high, medium, and low subsidy. So the subsidy has a context-dependent effect upon the pool, but that context is really dependent upon water residence time. Because if we graph water residence time here by number of hippos, the number of hippos does not matter to the degree of how the subsidy affects the pool. It's all about water residence time. If I break the pools up by their biogeochemistry, the high subsidy pools had really high biochemical oxygen demands, soluble reactive phosphorus, ammonium, total nitrogen, dissolved organic carbon, potassium, methane, carbon dioxide. The medium pools were really high in sulfates, and the low subsidy pools were high in nitrates. And so to break this down even more simply, the low subsidy pools remained oxic, they had oxygen, and the medium and the high were anoxic. And so this is basically the redox scale that you see here. If we look at the biochemical oxygen demand in high subsidy hippo pools, it is several orders of magnitude higher than untreated raw sewage here in the United States. So this is like a really extreme environment. If we look at dissolved methane, if this hippo pool was in the United States, it would be declared an explosive hazard, and you'd have to take remediation measures because all that methane coming off the pool, if it accumulates in some type of confined space, poof, it could blow up. It's crazy. And when you look at these high subsidy hippo pools, you can see it. This is methane bubbling from the bottom of the pool, like floating, decomposing hippo feces on the top. If we look at ammonium, a lot of fish could not survive directly in this hippo pool because the levels are just, crazy high. Same thing with hydrogen sulfide. Anybody who has fish in aquarium, like hydrogen sulfide will very quickly kill all your fish if it ever gets to that point. It's extremely toxic to lots of different types of fish. So for these really high levels to develop, we needed to figure out what was a vertical stratification in the pool. Some of these pools are only like a meter deep, or 2 meters deep. So it would have to be very, very vertically stratified for these really high levels to develop in the bottom waters. So we worked with our colleagues at Carnegie Mellon, and we built a robotic boat that I could pilot into the pools. And then at Yale, I developed a custom water quality sensors that I could do 3D profiles of the pool. And hippos are super dangerous, of course, which is why we had to use this boat with this sensor package. So here's a short video of it. This one has a little like crocodile's head on it, to try to distract the hippo, so he wouldn't like mess with it. So as you can see, the hippo doesn't really mind it. He's kind of cautious. He's like, is that really a crocodile? But in this hippo pool, there's usually about 100 hippos in it, but you can't see a lot because a lot of them are underwater. And so if you watch closely behind the boat, you'll see the rippling of the water come up here. There's a hippo underwater that hears the little motor inside, and he starts coming up on the boat back behind it. I mean, hippos are super dangerous, super curious too. So these are the 3D interpolations of pools with no hippos on top and a high subsidy hippo pool on the bottom. So again, this gave us really good evidence that we do have this a really high degree of vertical stratification within these high subsidy hippo pools. They're really extreme environments. So how do the hippo inputs interact with that context to shape the biogeochemistry? The high subsidy pools have just lots of reduced compounds. The medium subsidies have lots of sulfates, and the low subsidies are lots of nitrates. And you can kind of see this is the low subsidy pool here. And so you see a little algal bloom kind of around the hippos because that algae really responds quickly to those nutrient inputs. Importantly, water residence time and discharge are more important than hippo number in structuring how a pool responds to that subsidy. The reason why this is important is because there's talk of culling hippos in certain areas to improve water quality. But what this means is that it's actually better to try to preserve adequate flows rather than cull hippos, because it's really about the flows, not the hippos. What's cool about the system also is that we have these really localized rain events where you'll see the rain come in off the savanna and drop just a ton of water immediately that runs off the landscape and into the river, and that causes these floods that occur frequently. We call them flushing flow events. And we really wanted to understand, how do these flushing flow events shift pools between different states from this oxic and anoxic state? So we selected 4 hippo pools. These 2 are on the Intiac, and I denote the more downstream pool in a darker shade. These 2 pools are on the upper Talc, and again, the darker shade is a more downstream pool. And we did our whole suite of biogeochemical sampling in this, and we did it right after the first flushing flow until the next flushing flow occurred. And so these are water level readings. And so this gray area was a part of our intensive sampling. I did a PCA using all of those biogeochemical variables, and I overlaid an isocline of biochemical oxygen demand. And so this is very low BOD, and that is very high BOD over there. And now let me turn it on. So the days after flushing on the top tells you at what point in time that the biogeochemistry was. And so you can see that immediately after the flushing event, they're all kind of over here at very low BOD, but as time goes on, the BOD increases over time. And the downstream pool of the pairs always goes further and moves faster. So, really pointing to that cumulative effect of upstream pools feeding stuff in the downstream pools, and that is having a much larger effect. If we look at some of the other data from this, so this is from one of the pools, we had dissolved oxygen at the surface, at the bottom and downstream of the pool, and this is the water level. Each of the dashed lines is a different flushing event that came through. And a couple key points I want you to take away from this is, after the flushing event, there was a rapid return to anoxia. So this is, again, dissolved oxygen. Right after the flushing event, it would crash again. So these pools were never being completely flushed out from these events. And then there's also evidence for downstream algal blooms in some of this. You can see downstream, when you get those really wild swings in dissolved oxygen throughout the day, the diurnal cycle, you have algae responding. When you look at some of these pools, it makes a lot of sense. I mean, this is just downstream of one of these hippo pools. It's just so nutrient rich. The algae are just like sucking it all up. So how do they move in response? These flushing flows really do shift these pools between these states, and that shift happens very, very quickly. And the hippo number is an important determinant of the speed of state shift because of the loading. It's how much loading you have going into the pool will turn a pool quicker if they're in the same context. And there's a potential cumulative effect from the upstream pools on the rate of that change. So we all know that hippo subsidies can alter ecosystem function, but the biogeochemical cycles are really controlled by the microbes. And so we wanted to try to understand, like, how are the microbes involved in kind of these processes? And that made us kind of think, the hippos are loading so much feces, but the microbes in the feces, like, are they responding to these environments? Are they continuing to function in the environment? So I'll propose that, does the Hippo Gut Microbiome play a role? We know Hippo Gut microbiomes are continually colonized by the environment. That's how we get our microbiome. It comes in from around us and the environment. And there are distinct differences between environmental and gut microbiomes, and that's because the biochemical environment is different between the environment or in your gut. But what if you had a situation of where the gut and the environment kind of looked similarly, had a similar like low pH, anoxic, protected from UV light, temperature regulated? Could you actually get, successful transference of microbiota from the gut into the environment where the microbiota doesn't realize it's not in the gut anymore? And so would that microbiota then continue to function in the environment? This is kind of what we sought out to ask. And so using that same set of Hippo pools, we also collected samples for the microbial communities, the microbiomes. And what we did here is we collected samples. We then extracted the RNA and synthesized cDNA. So we wanted to know not just the communities that were there, whether they're dormant or active. We only wanted the functional communities, the ones that were transcribing. And so that's why we use the total RNA for that approach, because we wanted to tie the functionality of the nutrient cycles and the biogeochemistry to the microbes that were present. If we look at the Hippo Gut Microbiome, this is from 10 hippos, we can see that they're pretty much dominated by these top 4 phyla, and the firmicutes are the more dominant, the fermenters, dominant phyla within the Hippo Gut Microbiome. When we parse out those different types of hippo pools by how similar they are, we see something really interesting. So this is, doing all that sequencing and then calculating a Brake-Hurst dissimilarity matrix and then ordinating it with non-metric dimensional scaling. So what this shows you is that anything on this ordination, things that are more similar to one another are going to be close to each other. Things that are more dissimilar are further apart. So I'm going to show you a couple of these, so just keep that in mind. And so, these different types of hippo pools do cluster together because they're functioning more similarly than other pools. What's interesting is that how hippo feces is functioning in the gut of the hippo is actually functioning more similarly to the bottom of these high-subsidy hippo pools than the actual tributary that these hippo pools were part of. When we start to look at the taxa involved with that, we look at the different taxa associated with upstream of a hippo pool, an area free of hippo input, and compare that to the functional microbial communities at the bottom of a hippo pool or in the Hippo Gut Microbiome. We actually see 87 distinct taxa that are only found functioning in the bottom of a hippo pool or in the Hippo Gut Microbiome. And again, most of these taxa are the firmicutes, the fermenters. So we're having a lot of fermentation processes from the Hippo Gut Microbiome. Those microbiota move into the hippo pools and continue to functioning as an extension of the hippo gut. We did this also during those flushing flow events. And so I've denoted the flushing flow events as that dashed line again. So right after the first flushing flow event, we started taking the samples until the next flushing flow event. And now what this is we took all that, the sequencing data of the functional microbial communities, used the Bayesian logarithm to try to figure out what source it came from. Did it come from the upstream hippo pool communities, or did it come from the Hippo Gut Microbiome? And what we see is super interesting. Immediately after the first flushing flow comes through, most of the functioning community in the hippo pool is actually from upstream, which you would think, right? Because you just have a bunch of water come through and wash things out. But then as time goes on, every few days, we get more and more of the functional community beginning to look more and more like the Hippo Gut Microbiome. And I'll mention about the unknown. Those are just probably generalist anaerobic taxa from the ultimate soil microbial reservoir that could not be ascribed specifically from the Hippo Gut Microbiome or from the other source, the upstream communities. Probably they were just responding to the change in biochemical environment. If we look at this another way, so again, this is the Brake-Hurst dissimilarity matrix. Dots that are closer together function more similarly to one another. And it's over this same flushing flow. So let me animate it. So the hippo feces is here. And what we see is that the bottom of the hippo pool community and the upstream of a community immediately after a flushing flow look almost identical. So they're functioning identically, but then they diverge over time as more and more Hippo loading goes into that pool. And then the bottom of the Hippo pool starts to look more and more like the Hippo gut microbiome, starts to function more like the gut. And you see the upstream of the Hippo pool kind of bouncing around in multivariate space is probably just responding to the cessation of flows and the algal blooms from some of these nutrient-rich waters. Again, I'm a big experiment person, so we did an experiment at our bush camp in Kenya to try to parse out which taxa are really involved, or which biogeochemistry is involved with some of these changes. And so we devised an experiment where we took hippo feces, we used our pressure cooker to sterilize it. We don't have an autoclave. We then spiked sterilized hippo feces as an inert carbon substrate, so it's just inert food for microbes, into each of the bottles. We then took river water and put that in all the bottles too. So now we've inoculated it with a normal upstream microbial community. We then took fresh hippo feces and squeezed the liquid portion out of it. If you are imagining what that looks like, this is what it looks like. This is Gamey, our long-term field assistant, squeezing hippo feces into a bottle so that we could use that liquid portion. We took that liquid portion and sequentially filtered it so that we got the bacteria onto a 0.2 micron filter. So we've only got bacteria on the 0.2 micron filter. And then all the viruses, the bacteriophages, pass through into that viral filtrate. So we really wanted to know how bacteriophages may be involved too. There's a lot of good evidence that bacteriophages can really drive nutrient cycling in the ocean, but there hasn't been much work in aquatic systems. And typically feces has a very high viral load. So we wanted to investigate that as well. So we subjected half of that viral filtrate to a UV light. So the UV light will inactivate and kill the viruses. And then we spike that into this treatment. So this has live bacteria in it from the hippo gut, but no viruses. So we've separated that out. This one has bacteria plus the viruses, and then the normal control. So then we destructively sampled those in replicate over about a month. And so what did we find is that this is another breakers to some dissimilarity matrix coordinated with non-metric dimensional scaling. So dots that are closer to one another will be more similar in their functionality. And then numbers represent the date of sampling. So all of these treatments move right over time. And we see that the control looks, the microbial community functionality is very different than the two other treatments with the bacteria or the bacteria plus virus. And interestingly, the microbial community and the bacteria plus virus and bacteria one, they diverge at the last two sample points. So there is some type of bacteriophage or virus interaction with those gut microbes. When we look at the biogeochemical treatment differences, the bacteria plus control, pH, ferrous iron, biochemical oxygen, and hydrogen sulfide were significantly different between those, something that we would expect. Interestingly, pH and ferrous iron were significantly different between the bacteria and the bacteria plus virus. So the viruses are, we don't know if it's lysogeny or the lytic cycle or exactly what's going on, but they are having some kind of role in this, out of the hippo gut in the environment. And super interestingly, between the control and the bacteria plus virus treatment, there is no biogeochemical treatment differences. But you guys, the earlier figures show that there was large differences in the functionality of those communities. We're still really trying to understand that. It may be something to do with like functional redundancy of communities, or we're not sure. But key points to take away, these hippo gut microbial communities do facilitate the increased production of reduced compounds in the environment, kind of like what we're seeing out in the wild. And then the bacteriophages are suppressing the growth or activity of some of those hippo gut microbes. We're still really trying to tease that apart. So does the hippo gut microbiome play a role? I hope I convinced you that it does. We see that microbial communities in the high subsidy pools are more similar to the gut microbiome than the river. These flushing flows disrupt the communities, and we get increasing similarities between the high subsidy hippo pools and the hippo gut microbiome after the cessation of those flows. And then the experimental evidence about the bacteriophages, which we're still trying to figure out how to interpret that. We've got some experiments planned to kind of really do some detailed work on that here. And so that kind of brings you to this idea, the metagut hypothesis. And I'll hat tip Matt Cohen, who coined the term at a conference a couple years back when we were talking about it. We know animal guts are continually colonized by the environment, and microbial community coalescence is just a result of an environmental filter or biotic or abiotic interactions. But if you can remove the environmental filter by the two patches having more similar biochemical environments, and remove the biotic interactions just by tons of loading, constantly putting new microbes in from the gut, we propose that, then you can get this kind of meta gut where these hippos in these pools are sharing this communal microbiome within the pool, and that's really driving some of these patterns and really interesting processes we see in the Mara River. We received some more funding from NSF lately to do some very detailed experiments to incorporate metagenomics, metatranscriptomics, and metabolomics, and we're doing this all over in the experimental stream array, just in the parking lot back over here. It was really exciting because we were able to hire a bunch of undergrads to help out with it, and Disney was really excited, and so we could get fresh hippo feces. So like every 4 days, Disney would bring hippo feces up right after the morning movement, and then participate in the experiment with us. It was really cool. But we're still working through a lot of the data, so I don't have anything to share on that yet. But we've got some really cool effects from it. And so really, I'm just proposing that animals are just living bioreactions, moving anaerobic bacteria across space, but then it's the context that they're putting that microbes in that can then alter ecosystem function on a scale that we haven't seen before, that we don't understand. So if I take a step back for a minute and I ask you to imagine like a body of water that's clear anthropogenic impact, not impacted by humans, you might think of something like this. You could go drink from it, swim around in it, and it's really nice and peaceful. But I would argue that the Mara River is actually what a river, probably most rivers, used to look like in the past. With just tons and tons of animals and microbes and feces and everything. And the Mara River, so for us, the Mara River is a really great place to study some of these patterns and processes about how animals can influence ecosystem functioning, either whether it be through subsidies or the subsidy of microbes, and then how those microbes can change the functioning within the river. So with that, let me know if you have any questions. I'm happy to talk about it. We have a lot of fun studying these things in Kenya, and we're doing a lot of studying here now too at the Disney Hippo. So if anybody's ever wants to get involved, just reach out. Thanks.
Questions in the room for Dr. Dutt?
I have a quick question. So based on your talk, it seemed like the hippo's microbiome is highly influenced by their environment. And so you'd expect hippos in Disney versus other places that you're studying to have different microbiomes, and what would be driving those differences?
Yeah, no, absolutely. That's something that we're just starting to look at. The microbiomes, differences in microbiomes between the captive at Disney and the wild population. Why I don't have that information yet, so we did a bunch of 60S sequencing of the total RNA in the bush out there, but with all the new captive stuff, we're using better technology. So we're doing shotgun metagenomics to get all of the microbes and all of the viruses and stuff like that. And so I have a bunch of fresh samples collected from Kenya in the -80. I need to do the shotgun metagenomics on it to do a proper comparison. And because one of the other issues with the methods that we use to do this in the field is we took the total RNA, which includes the messenger RNA and the ribosomal RNA, which does not always correspond one-to-one with functionality. It gets us close, but it's not. That's why we're doing the metatranscriptomics over here, which is only using messenger RNA. Because like RNA, if you look at it funny, it disappears. It's like super ephemeral. And so we needed to do most of that stuff here, where we could have the lab space and the clean conditions and stuff to really get at that. And so, but it's super interesting. I'm going to the American Zoological Association convention next month to pitch an idea to zoos about kind of that question, because some of these captive hippos have like irritable bowel disorders and things like that. And each of these different zoos in the US who have hippos have different types of filtration systems for the water, right? They filter the water because they want the tourists, people to be able to see them under the water. Otherwise, like a natural hippo pool, you can't see in that water. But that's how they evolved, And so it's interesting. There's a lot of really interesting ideas there about that, the differences in the microbiome. Because also here they give them like a bunch of vitamins and, you know, things like that, which is different than the wild population. Yeah, it'll be interesting once we can get the time to sequence that. Yes.
I was just thinking about the fact that perhaps this phenomenon existed, but no longer does, but there may be a perpetual scale. That one slide that you showed looked like tadpoles. And so over a seasonal basis, you have tremendous numbers of tadpoles clustering, then they go off as frogs and then come back again as tadpoles. So there may not be the buildup that you have, but there may be something like that.
Yeah, no, that's super fascinating because we thought about it like, you know, this whole idea, like where could we find it here, you know, in different systems, you know, like beaver ponds, you know, beaver ponds on the bottom go anoxic, and you've got cohabitating animals in aquatic environments. As long as we can get it, as long as the environment goes anoxic, then you can start to get close to the conditions of a gut of an animal. But even then, you know, like frogs and beavers and stuff, their guts probably are not as anoxic as, or low pH or acidic as ours or hippos. And so it's super interesting. We've also thought about looking at this in the Galapagos, with the Galapagos tortoises. Because if you're familiar with what they do, down by the ocean at the bottom of these islands, they like hang out in these big ponds of really muddy, nasty water, like all the hippos, and then they like get out and slowly walk up the hill, eat a little bit, and come back and like bask in these like communal, like, you know, pools essentially. So we think we might see something there as well. It's definitely something that we're always thinking about. So if you guys have ideas, let us know. We can go look at it together.
Any other questions in the room? It's like, I don't know how severe like the hippo population in Colombia is because of Escobar and that, but like what would be the impact on the ecosystem there?
Yeah, it's a good question. And that's a question I'm frequently asked. You know, it's a really fascinating natural history experiment, I guess you could call it that. You know, what's going on in Colombia. Are you guys familiar with that? About Pablo Escobar's hippos? When the army took over, they let all the hippos loose because they're too hard to control, and there was like 6 to 8 hippos decades ago, and now there's like between 40 to 100, and they've gotten out into the Magdalena River. So Magdalena is like a beautiful river. It's very, very large. You know, we went down there in 2018 to kind of work with some colleagues on kind of some of those questions. I don't know if we get these same effects until we get more hippos, you know, because like the Magdalena is huge and the hippos are really dispersing out, you know, there's so much habitat for them in the Magdalena. And interestingly, humans are helping them disperse because when humans find a baby hippo, they're really, really cute. And so they'll sell them to a cousin or a friend like 100 kilometers away on the other side of Colombia. And then all of a sudden you get like this other group of hippos show up. It's like a really interesting thing. But until we get the context dependency of these high subsidy pools, you know, in Kenya, it's like a semi-arid environment. And so you can get these really high concentrations in a very low quantity of water. And that's what really drives it. We don't see that happening in Colombia yet. And it might take a while for us to start to see something like that there. Although one thing I didn't talk about was, you know, the fish and the invertebrates, they eat hippo feces. It's like a really good food resource for them. And so there are probably fish and inverts in the Magdalena that are benefiting from that food resource as well, and maybe providing some type of you know, nutritional advantage for them. Because, you know, as part of this whole communal gut microbiome system, we're also looking at it within the fish and the invertebrates, since they consume hippo feces. So do the fish just get really, really big in the Mar River because they eat a lot of hippo feces, or are they actually assimilating gut taxa from the gut microbiome of the hippos that then continue to break down that hippo feces in their gut, in the fish gut, that provides them some type of physiological advantage? So we're kind of looking at that as well to try to understand that. So that could be happening, you know, in the Magdalena.
So the same fish is getting adapted to the anoxic conditions as well?
I'm sorry?
The fishes that you just talked about, are they also getting acclimatized to these anoxic conditions?
Yeah, it's interesting. So there's a number of species in that river, in that general area that can tolerate hypoxia, very low oxygen. They can gasp, they can air breathe, some of the catfish, and we have lungfish there as well. Which is why I don't think it's the anoxia that's killing the fish. I think it's the hydrogen sulfide or some of the reduced compounds that kind of come through these events. But what's really interesting is like hydrogen sulfide and other reduced compounds like that they rapidly oxidize, especially when a big flood comes through and you've got all that turbulence, really increasing the rate of aeration of that pool. And so only at certain points in that flood pulse will you actually have a really high level of hydrogen sulfide or some of those other reduced compounds before it's oxidized enough, and then the DO starts to come up. But it's where that, it's a difference of thinking about a static water quality meter where you see the crash in DO happen and it comes back up. Well, do we know that was at the peak of the crash in the river system, or did it actually happen 10 kilometers up and the whole thing was anoxic for 10 kilometers and then it just started to come back up? Do you know what I mean? It's a really, conceptually, it's very hard for me to like understand and talk about it, which is why I haven't like really figured out how to model that. Yeah, but that's one of the issues as well. So not all the fish in the river are going to be wiped out across the whole flood pulse, because you're only going to get that crash that really hurts them at a certain spot in the river network before it's oxidized and the DO starts to come back up and you get dilution of the suspended sediments and stuff.
I don't see any questions online, so anything else in the room? Okay, one last round of applause, please.
Great, thank you very much. Yeah.
Thanks.
You did something.